So far we have seen those complicated algorithms, and we have seen those their algorithm executions. So don't worry, we have another cool representation method. So here we are not talking about algorithms, we are simply placing two strands with one another, and we are comparing two nucleotides or two amino acids in these strands, and then we are putting some symbols, like whenever we see a match we put a cross or star or something like that. So in the end we come up with some graphic representation, and we can get some interesting patterns out of it. Basically this arrangement is called as dot plots. So dot plots, they show intra sequence comparisons. Sometimes we take the same sequence and we try to find out whether we have those repeats in that sequence, or how many repeats we have, are they direct repeats or invert repeats. So we'll do that and see that in a moment. So if we have some similar identical sequences, two sequences which are very similar, they are plotted together, so we can get a diagonal line. So diagonal is where we get the same nucleotides on both the sides at same location. So in this way we will get that diagonals. We do use these things a lot. There is an algorithm coming in the next section we call as FASTA. So over there it is used. Same way, if we take two genome assemblies, we can compare that while using some techniques like this. So that's simple and easy but cool. So here we have this sequence is written as sequence analysis primer. So we have the same sequence on both the sides. So what you see in the middle is the diagonal. So where we have the exact, these exact sequences. So we have the diagonal. So that is, if two similar sequences are placed, we will get those diagonals. So that diagonals tell us the sequences are similar. Here we are placing the same sequence, and then we can, in addition to that, we can derive some other interesting patterns. We can see some lines going parallel to the diagonals. What are these? Some lines going perpendiculars, and maybe we might find later on at some stages we might find some lines like this. So everybody has different meanings. So let's see what we can get out of it. So it shows mutated inter-sequence comparisons. Jump or shift in the main diagonal of dot plot points out the existence of indels. So whenever we have gaps or indels or shifts in the diagonals, we can say that we have indels in it. So you will understand while just looking into it. So over here we have one sequence that has sequence analysis primer. On the other side we have sequence primer. So diagonal was going like this, and then there is a gap. So this was the full length, and over here this area tells us that there is some deletions. Insertion on one side, you see the word analysis is missing in others. So this is one thing which we can get out of it. Another phenomena that is easy to visualize with dot matrix are duplications and direct repeats. So duplications are seen as distinct columns of diagonals. So let's see how do they look like. A row or column of diagonals in a dot plot shows the direct repeats. So let's see. So we have sequence analysis primer, and over here we have sequence, sequence, sequence. We see this, this, this. What we have? So we have direct repeats. Same way, we can look into the occurrence of some elements over there. So how many times we see those E's in the other one? We can see over here. And for individual nucleotides, we can also look into their occurrences. So many times we see a lot of this letter E. So if a diagonal, there are more than one diagonals in a dot plot in the same region, we can see that there are more repeats in that region. So here is how inversions they look like. So if something is inverted and then in one of the strand it is in X to Y direction, and in other one it is flipped around. So this is how they look like. These sequences are shown as contrary to the diagonals, showing that similarity. Low complexity regions, they can be found as the regions around diagonal, all obtaining a high score. So square shows a low complexity region. So here, those regions where we are not sure what's going on, so they look like this. So in this section, we observed that using those simple techniques like dot plots, they can be really useful in getting some evolutionary relationships between two sequences.